It's not showing, but my face is not showing, but that's okay. Okay, everyone, it's good to see you, even if it's just digitally. I'm going to talk to you today about some fossil aroid spadices. Aroids are fossil or flowering plants in the family Araceae. They are characterized by the spathe and spadix. You see here in this glass, the spathe is a modified leaf, which can be variously colored. And the spadix is the inflorescence axis on which there are a large number of flowers. Damn, okay. Over on the right-hand side, you can see Lysochaeton, which is the Western skunk cabbage. And you see here the spathe is yellow in this particular species. And the spadix, which here shows the peduncle, and then the zone of bisexual flowers. So there are bisexual flowers, meaning they have stamens and pistils, and they are helically arranged. I'm going to be talking about 3 different spadices today. They come from Alberta and British Columbia. First of these is Albert Arum poirei, which comes from the Campanian Upper Cretaceous of Alberta. The second one, Bognerospatix spearsii, which is associated with a leaf called Orontiophyllum grandifolium. This paper is in press. I just read proof, and it should be out maybe next week. This is from Alberta, from the Blind Man locality, Paleocene in age. And then the Appian Way spadix, which we can move now into Paleocene. Perhaps the plants at Appian Way are very similar to Eocene at Clarno nut beds and in the British London Clay Formation. Okay, Albert Arum poirei. This one is described by Bogner, Hoffman, and Allenbach in 2005. It's from the Late Cretaceous Horseshoe Canyon Formation. And you'll notice that the shape of this spadix is not elongate like we've been looking at already, but it's kind of globular in shape. Each one of these things is a flower, and a close-up of one of the flowers on the lower left. You can see the style, which is sticking up. You can see that in the side view here. And the style is angular, which is very characteristic of this flower. And the stigmatic surface, which is right near the arrow tip there, is the place where the pollen would have landed. Now, these flowers have tepals. Stefan talked about tepals, where they have the sepals and the petals can't be distinguished from one another, and they may be the same color. We usually use the term tepal. So there are 2 whorls of tepals in this spadix. You'll notice on the back side of this specimen, and this was preserved in silica, and so it is preserved in 3 dimension, and we get internal detail. On the back side of the specimen, you can see these clusters of 3 seeds, which we'll look at a little bit later. Now, the peduncle would have been located here at the bottom. A closer look at the seeds, you can see they're in clusters of 3 there, and they're actually, this is one fruit, so there's a cross-section of the fruit showing the seeds inside. There's 1 seed per locule, and there are 3 carpels in the fruit. So you can see the outline of the carpel here and the seed inside. The seeds look to be kind of spiny, but when you actually look at the fractured surface, it turns out that they're striated and a little bit curved. These authors compared the inflorescence or the spadix of Albert Arum to Symplocarpus. This is Eastern skunk cabbage. Now, what I've illustrated on the upper left is just the spadix itself. The yellow stuff are pollen sacs, so the stamens are sticking out, and then we have the tepals here on either side. Over on the right-hand side, this is more easily seen. This is a little bit further along in the fruiting stage, but we have 4 tepals, so 4 parted, and then an angular style. It's very angular. This, on the bottom in the center is what the spathe looks like in the Eastern skunk cabbage. It was compared to Symphocarpus, but you'll notice there's some differences. Symphocarpus, there's 4 tepals in each flower, and Albertarum, there are 6. There's 1 locule in the fruit in Symphocarpus and 3 locules in the fruit in Albertarum. There's 1 seed in each fruit only in Symphocarpus and 1 seed per locule in Albertarum. The seeds in Symphocarpus are globose and smooth. The seeds are striate and curved in Albertarum. The second spadix type is this one, the Bogner spadix. It's cylindrical and more elongate again. It's at least 4 centimeters long, but we're missing the apex and the base. It's about 1 centimeter wide, and there are numerous helically arranged flowers. You can see that pretty well on both of these specimens. What we have on the left are the part and counterpart of the holotype, and this side has the carbon, so there's the compression and then the impression, and you see the same flowers on each side. But we do have permineralized seeds and partially permineralized fruits. So this is a paper that should be coming out next week. Oh. Flowers are varigoniate. That is, they have petals or tepals. In this case, we have 6 tepals that are hooded or bent over, and they're in 2 whorls. You see these T1 and T2. So 1, 2, 3, the outer whorl, 1, 2, 3, that's the lower one. And you can see the counterpart on the other side. The arrow here points to a seed. We'll come back to those. Now, did these flowers have stamens or not? Are they bisexual or not? We have the seeds, but do we have the pollen? We have no pollen preserved, but there is some indication that we might have stamens. So we believe they're bisexual for a couple of reasons. If you look at the picture on the right-hand side, you'll see there's Pothos, a living aroid, described by Busco in 2001. And this also has 6 tepals per flower. So you see the T1 and T2 whorls. In the center of that flower is the pistil. It doesn't have as an elongated style as we have in the fossils. But here are the anthers, the pollen sacs. And you see how very filmy they look. These could have rubbed off, brushed off, fallen off as this thing was washed into water and deposited. In the white circles in B and C, we have some scars left over from places where maybe the stamens have fallen off. But I think I have better evidence in the next slide. Okay, on the left-hand side, C and D are, again, mirror images, but this shows the flask shape of the pistil. So the pistil is a flask shape, and same one on this side here. The arrow points to a seed. And notice that these are longitudinal sections of flowers in E, F, G, H, where we have the tepal labeled T1 here. You'll see it's hooded, it's bent over, and it covers over the base of the pistil, but the stigma sticks up through the tepals, and there's a lobed, the style sticks up through the tepals, and there's a lobed stigma. So the style is located with the white arrows on the side, and the red arrowhead, you see some black sort of filmy stuff here, kind of elongate structure, which we think might be actually stamens, but we have no pollen. In G is a pretty good view, a side view of that pistil, these flash-shaped pistils, and these also have the seeds in place. You can see the seed in place here. In H, you also see the style sticking up through the hooded tepal. There's 3 carpels in this fruit, and they have axial placentation and 1 seed per locule. Okay, so this is a lucky break in J, where we can see a cross-section through the ovary, and you'll see there's 3 locules. So this is 1 fruit with 3 locules, and then this is the placenta here. So we have axial placentation. The seeds are borne along the axis. Similar thing that you see here to Pothos in K, described by Ide in 1967. Now there are quite a few seeds in some parts of the specimen, but we couldn't get much closer with the dissecting microscope. So what we see is what we get. But you'll see that the seed coat is quite thick. And you also see the seed is at a regular shape. So it has one small end and one fat end. The narrow end is the micropolar end, the fat end is the chalazal end, and the outer surface of the seed is varicose, it's kind of bumpy. This compares very favorably to Spathiphyllum lancifolium. It's an aroid that's described by L.T. Seubert in 1993. So we have a narrow micropolar end and a wide chalazal end with a lot of bumps on it and striations. We also see, this is the raphe, the vascular tissue to the seed, which we can't really make out in these, but it may be visible there at that blue arrow. When you look at the integuments of these seeds, you'll see they're quite thick, and they could be quite similar to what's seen in Spathiphyllum as well. This is our reconstruction of the Bogner spadix flower. Looking at it from the outside on the left, you see the two whorls of three tepals, outer and the inner whorl, and then the lobed stigma. And that style, which sticks up from underneath the hooded tepals, stigmatic surfaces up here. And then we have one large seed in each locule. The Bogner spadix spadices are associated with odontioid leaves, genus Odontophyllum, this particular leaf, Odontophyllum grandifolium. And we think these probably represent parts of the same plant because at this locality, we have one type of aroid spadix and one type of aroid leaf, but they're not attached. So they have different names at this point. We'll come back to what we did with these later. This is the third type. This is the Appian Way spadix. And for comparative purposes, I put Lysocheton in here as well, the living Western skunk cabbage. This is the longitudinal section through the spadix, which you see here in these two figures on the left. We think we have a peduncle, which is 14.5 millimeters long, but it's pyretized, so it's not really well preserved. The apex of this is missing, but we have a lot of helically arranged flowers, many helically arranged flowers. The section right through the center, like would be this one here, we have a wide pith, but you'll see there are some sclerenchyma partitions here in the fossil, which we don't have in the living stuff. The second specimen we have is a cross section of the same particular type. These are about 37 millimeters long and 8 millimeters wide. In this slide, we show the tepals and the pistils and the potential stamens. There's been no fossil pollen found in any of the material. A longitudinal section up at the top showing a large number of flowers. And then we have red arrows pointing to the hooded tepals, which you can see pretty well here on this specimen and this one. There's another one in the center. And the black arrow pointing to the style, which you see here up in the upper register up here. The style sticks up, but it doesn't really extend way out beyond the tepals as it did in the Bogner spadix. And then we have some remains in between these petals or tepals and the carpels, and these long stringy things here might be remains of stamens, we're not sure. There are 3 carpels in each of the fruits. You can see them here and better seen in the upper right. 3 carpels per fruit, 1 seed per carpel. They're not that well preserved, but you can see that they don't have a lot of spiky protrusions or anything on them. And this is the main axis of the fruit. This is a cross section of the fruit. The main axis of the fruit is here. If you took a section through, you would see the axis coming down here. It's not very broad, but we think we have the attachment here, and they're probably basally axile placentation. And the seed you see is kind of ovoid shape. Now, what we wanted to do is to discover where these would fit within the Araceae. And what we used was 83 morphological characters. This is a majority rule consensus tree based on modified Cusimano et al. Morphological matrix. This was updated in 2019 by Sender et al. And this shows you the subfamilies of Araceae. So the whole Araceae is in here. Acorus and Tophieldia were used as outgroups. This was just the morphological tree. And you'll notice this group here, Lemna, Spirodella, Wolfia. These are the lemnoids. Some people include those as a separate family, Lemnaceae. So these are duckweeds, which usually using DNA fall into the family Araceae and not outside of it. But morphologically, they're basal. I wanted to point out this group, the Proto-Araceae, Aurantium gymnastacus, Lysocheton, and Symplocarpus. We looked at the two skunk cabbages, eastern and western skunk cabbages already. Then below that is Calla, and then these groups, Apothoideae, Monsteroideae, Lazioideae. These are all bisexual aroids. And then below this is a very large group of unisexual flowered clade. So we have portions of the spadix, which are male, and parts that are female, if you will. So these are what the bisexual aroids look like. So all of the flowers or fruits are the same from the base to the apex, which is what we think we have in our fossils. Okay, what we got when we put these into, we have 96 taxa, 83 characters. This is the strict consensus tree. And what we have are the fossils are in red, and the spadices and the associated leaves, which we keyed in with Bongor spadix spearsii, each conform to an early diverging lineage of Araceae. So these are quite early on in Araceae, which is increasing our knowledge of the Proto-Araceae. And they provide strong evidence for extinct Proto-Araceae. These have a novel combination of characters during the Cretaceous and the Paleogene. We're going to really need more fossils to get a clearer picture of how the changes occurred in the evolution of modern aroids. But for now, what we have are proto-aroids, both in Cretaceous and Paleogene. Thank you.
Okay, are there any questions?